Okay, so this is going to be the last lecture. Here, what we are going to see the second part of the course that we were talking about the signals that are coming from the body, right? So we have already seen how the cell is basically acts like a source of electrical signals by looking at the action potentials, right? So the cell are basically at a resting membrane potential, which is roughly around minus 90 millivolt. That means between the interior of the cell and the outside cell, the extra or intracellular regions, there is a potential difference of minus 90 millivolt, right? And due to some disturbance, this minus 90 millivolt can increase. The cell potential inside the cell, the potential can increase to an order of say minus 40 to minus 60 millivolt. Then what happens is the sodium gates can open, right? The activation gate of the sodium will open from outside. Sodium will start coming inside. But then we found that at the same time, a signal is given to the potassium gate to open so that the potassium can go out. But then that will happen only from a given with a given delay, right? So by the time a lot of sodium will come inside, that will increase the potential of the cell from minus 90 to plus 35. That means cell interior of the cell become more positive, that we have seen, right? But then the potassium channel will open and it will go out. At the same time, the sodium channel is deactivated. So it's an inactivation gate. So sodium will stop coming in, whereas the potassium will start going out, and that causes the potential again go back from plus 35 millivolt to minus 90 millivolt, right? So the process of increasing the potential of the cell from minus 90 to plus 35 by the sodium channel is called the depolarization, and the retrieval of this resting membrane potential from the peak to the resting membrane potential is called the repolarization, which is initiated by the potassium channel, right? So this we have already seen, and this inactivation gate will be closed till the cell membrane potential reaches minus 90 millivolt. Once it reaches, this guy will be open, the activation gate will be closed, so everything is now under equilibrium. So that's how the action potentials are being generated. Now we'll see some certain special cells called the neuron cells, which form the basics of nervous system. So essentially, this acts as like a sensors to the environment. So we have already seen that it's important to have homeostasis. So in order to have homeostasis, one important thing is to have a, basically it's a form of a feedback, but feedback requires sensors, right? That you have already seen in the first few lectures. So essentially, these sensors are required. That means we have to see the world around you. You have to basically visually see it. You have to basically physically sense it, like skin, using the skin. You have to hear the sound. You have to sense the temperature. You have to hear the sense the pressure. So there are many things that is around you which is kept optimal for our living existence of the living beings. But if it is perturbed due to some reason, so for example, when you are in a very cold country without proper clothing, then there are sensors so that you have to take an action. You have to basically run to a shelter, right? So you have to have some arrangement for the cloth. So that's a part of homeostasis. And who is doing all this stuff? By our own sensors. So these sensors are basically the neurons. So the basic structure is the neurons. Basically, it's like a the cell structure is shown here. There are different types of neurons, but one cell structure is shown here. So it has got a soma, which is cell body, and then it has got a very elongated structure called axon. And there are several small branches from the soma, which is called the dendrites. Now you can view it like a input. This dendrites as the input. So the data from the input or from the other neurons will come inside this through the dendrites. It will be processed in the cell and then it is passed through the axon to what are called the axon terminals, from which this can be transferred to the next neuron, right? So the external disturbance that I was telling you for the why this action potential is being generated, basically it can be because of the sense. When you touch, that can create an external disturbance which can cause imbalance, and that's how we sense it, right? So these are basically the electrical signals being responsive to some external disturbance, which is how we sense the sensation of touch. So here you can see that suppose you have two neurons, of this form. So neuron has got a soma and dendrites. Dendrites acts as the input. And then you have got a axon, and then axon has got several branches, and it acts as the output. So an action potential which is being received from the input end can pass through the axon, and it can go through the axon terminals, and it can go to the next neuron like this. So a dendrite receives the input to the first neuron, like what you have shown here. It will pass through the axon to what are called these myelinated axon trunks. So this myelinated sheet makes sure that this action potential travels very fast. So we already seen that how action potentials travel. That is there in one of the lectures. In fact, in the previous lecture. So essentially, that this myelinated sheet makes sure that it moves very fast, and then it reaches to the axon terminal, and it can be connected to the next dendrite of the next neuron, and that's how the connections happens. Now the question is, how do you connect them? So essentially, to connect them, what we need, it is not a physical connections, because there are several that's called the neural networks. Essentially, there are several neurons being connected to each other. So more than one can be connected to one or the another neuron, right? So a physical connection is not a something which is what you call it judicious, because it requires some spatial, and it's a rigid structure, right? So there should be some way of wiring and rewiring. That flexibility should be there. So for that matter, this type of connection between these two neurons are not actually physical connection, but or it's not a continuous system, but it's basically discrete neurons, but they are communicating each other through a certain mechanism. So what is that mechanism is what we are going to look at a little bit in a very, very quick way. But if you want to learn this subject very well, what I suggest you is to look at our department course list. There are two courses in the department. One is offered by Professor Rohit Manchanda. He takes this subject in very deep manner. And there is also another course offered by Professor Neeta Kanekar. So you can look at both of the courses and decide if you want to, if you want to learn more about it, or look at it in a more deeper way. So I'm just going to give you some idea about what is going on. That's all I can do. So here, so the idea is that these two neurons, basically, they are connected to each other, not physically, but they are kept close to each other. So this particular dendrites, that is axon terminals, that is coming from the first neuron, is kept very close to the dendrites of the next neuron, very close to each other, so that there is a small gap. And from there is a communication happening between these two neurons in a special way. That's what we are going to look at. If you look at this left figure, you can see that there is an axon terminals here from the first neuron, which is kept very close to the second neuron's dendrites. And there is a gap here. So there is no physical connection here. Now what happens is this particular axon terminal of the first neuron is called the presynaptic terminal. This particular area, which the gap, is called the synaptic cleft or synapse, and the dendrites of the next neuron is called the postsynaptic neuron. So it's the presynaptic terminal, postsynaptic terminal, and the synaptic cleft, right? And the communication happens through this cleft in a way which we are going to explain. So what happens when when an action potential is picked up by this dendrite, and it is passing through this myelinated sheet, which is through the axon to the axon terminal. So when this action potential passes through this axon terminal, basically what you know, that a lot of sodium ions are going inside, some of the potassiums are coming out. So there's a depolarization and repolarization happening, right? So in such case, a lot of sodium and potassium, a lot of sodium ions are being generated because of this action potentials, and along with that, there are some calcium ions also being generated. So in neuron cell physiology, basically there are some it's not just the sodium ions that are involved in creating the depolarization, but it is also contributed by some calcium channels as well. So that's what I said. We are interested in basically the sodium, potassium, and the calcium. So it's true for the heart muscles as well. So calcium is also taking part a lot of role role in neuron signaling as well as in the heart, how heart pumps, right? So it also requires some electrical signals. There also the calcium channels are very important. So here both the sodium and the calciums will be present in the cell in the presynaptic terminal because of the action potential, and this basically will actually causes certain release of what are called the neuron transmitters. So here the idea is like this: there is an electrical signal coming from this particular axon terminal, which is the action potential. Now this action potential will terminate here, and this action, because of this action potential, certain chemicals are released here. That chemicals will pass through the synaptic cleft, and then it will bind to certain receptors here, and that causes an action potential to happen in the next neuron. In fact, there are two possibilities. One is these receptors can pass this action potential to the next neuron, or it need not pass this action potential to the next neuron. Both can happen. But the idea is that an electrical connection is now converted to some sort of a chemical connection through some chemical reactions, and then it again passes through electrical. So it's basically decoupling the electrical connections by using some chemical trajectory. That's what it do. So why is it so? Why do you need that? Why not to connect it rigidly? Why do you need a chemical connection here? What is the role of neurotransmitter? Why a neuron need not be a continuous structure? So all these questions you have to ask yourself, and a nice course on electrical physiology basically tells you all these answers. That's what I told you to look at some dedicated course for this. So many questions can be answered. In fact, at some point, people were actually not knowing that there is this type of chemical reactions are happening for these connections. So it was invented, of course, long ago, but people were ignorant about it. And then they asked this question. Once they discovered it, they asked this question: why do you want it? In fact, right? So these are the things that you get to think. So the idea is that there are some certain things called neurotransmitters, which are actually put it in some sort of a capsules called vesicles. So vesicles are some sort of a carriers which is present inside the cell. You can see it in normal cells where basically its role is to catch the intruders like bacteria and all, and then try to destroy it. So the vesicles are present inside the cells. So these particular vesicles has got a lot in the neuron, has got a lot of neurotransmitters being synthesized using energy. That means basically from ATP to ADP conversion. And in order to transmit this action potential from the presynaptic neuron to postsynaptic neuron, basically you need energy because you need a lot of neurotransmitters to do this job. So you have to synthesize it a lot, and the mitochondria is again helping to synthesize these neurotransmitters, right? So when the calcium and the sodium ions are present as axon terminals gets this action potential, these neurotransmitters will be released to the synaptic cleft, and they will start going out to the next postsynaptic terminal. In the postsynaptic terminal, it can get bind to certain receptors, right? So postsynaptic terminal is nothing but some dendrites of the next neuron, like here, like what I have shown here, this side. So the output of the one is going to the input of the next neuron. Now, so as I told you, there can be two types of ways in which cell can communicate via neurotransmitters, which is basically what are called the ligand initiated gate opening that I have told you in the beginning when we were talking about how a gate can be opened in the cell membrane. So we told a sodium channel or potassium channel, mostly these are opened using the potential differential between the cell and the outside, inside and the outside. But it can also be initiated by certain materials when it get attached to the gate. They can take a conformal change of the conformational change of the protein, and it can also open and close the gate. That can also happen. So here, in for case of neurotransmitters, basically it's a ligand gated initiated gate opening. The other can also happen. Voltage gated channels can also actually happen because actually the ions can actually cross the membrane and say move through the synaptic cleft and initiate some gate opening or closing in the postsynaptic terminal. That can also happen, but mostly for neurosignaling, it's a neurotransmitter is what we are looking at. This ligand initiated channel opening is what we are looking at. So here, coming back again to this problem, so you can see that action potentials generate the calcium and the sodium. Calcium ions will initiate the release of neurotransmitters from the vesicles. So it's a role of calcium to release the neurotransmitters from the vesicles. And these neurotransmitters will move through the synaptic cleft and it will get attached to the receptors in the dendrite. Now, this particular neurotransmitters, there are different types of neurotransmitters. Depending on the nature, two things can happen. One is excitatory postsynaptic potential. So excitatory postsynaptic potential means if my neurotransmitter is of the nature excitatory, that means it initiates excitatory postsynaptic potential, then it means that when such an neurotransmitter gets attached to the dendrite of the next neuron, essentially it will initiate either the sodium channels to open so that a lot of sodium will go inside the next neuron cell, or it can also initiate a lot of a lot of calcium ions to go. So essentially in both cases, a depolarization of the next neuron will happen so that an action potential will be created here. That is why it is called excitatory. So an action potential here is passed through the neurotransmitter, and if the neurotransmitter is excitatory nature, it will cause sodium and calcium ions channels to open in the next neuron so that another action potential will be created here so that this action potential in some sense is passed to the next neuron. Right? So that is called the excitatory postsynaptic potential. For example, glutamate is such an example of a neurotransmitter which creates this EPSP. Another option is this particular neurotransmitter from the first neuron, it reaches and get binded to the receptor in the second neuron, but then it causes inhibitory postsynaptic potential. So what do you mean by inhibitory? So the idea is that these particular neurotransmitters will cause two things. It can cause two things. One, it can open the potassium channels of this cell. But you know that potassium channels, when potassium channels are open, potassium is abundant inside the cell. So by simple diffusion, all this potassium will go out from interior of the cell to outside. So that will causes the resting membrane potential of the cell to go beyond minus 90 millivolts. It can go much lower than that. Or this receptor can also cause chloride ions to go from outside to inside. So when chloride ions move from outside to inside, because they are negative ions, basically it also decreases the potential of the cell. So basically it is minus 90 millivolt, but this process can take it to minus 95 millivolts, say for example. It's become more negative from the resting membrane potential. So if you want to depolarize it again, you need more and more sodium ions, right? It's become it's difficult, and that process is called hyperpolarization. So in some sense, it means that we are inhibiting this postsynaptic neuron so that it will never take any action potentials. The signal cannot be passed. So that is called the inhibitory postsynaptic potential. So these are the two things that can happen. So there are so many other details. I'm not going to those details, but what we are going to look at is as we agreed that we are going to look at the signals that are generating from the body. So we were trying to understand two such signals. One is the EEG and another is the ECG. So EEG is what you are going to look at. How do you relate all these things into EEG? So if you look at this EEG, so EEG is something that is being picked up from the brain, from the scalp, right? So we put a lot of electrodes in the human scalp, and these scalps are basically some electrodes in sort of in a jelly like which acts like an electrolyte, right? So then the electrical signals, if there is an electrical signal in the scalp, that will cause the redistribution of ions across this electrode, which can be measured, which is manifested as a EEG signals that you might have seen in the hospitals, right? Now, from where these electrical fields actually comes in the brain, in the scalp. So that's what we are going to look at. So essentially, these are caused by special neurons which called a special neurons which has got a pyramidal structure. So these neurons are basically put in the cortical region. So if you see this brain, if you look at this brain, so you can see there's a scalp at the surface and there's a skull which is bone basically. And then then you have the brain tissue. And the brain tissue at the surface is basically the cortex part. In that cortical region, there are a lot of neurons being kept, but you can see the dendrites are a little bit elongated for this type of special neurons with pyramidal structure, and they are kept in a very nice fashion. So for example, they are stacked parallelly. All the dendrites are facing up towards the scalp, and there are many such neurons are being packed or stacked parallelly. So in the same direction. So it's vertically from the scalp to bottom, but dendrites are towards the scalp, directed towards the scalp. That's how it is being stacked. So this is the structure of the brain, and this is exactly why we are actually getting nice EEG signals at the surface. So if you look at the structure of this particular neuron, you can see that there is a dendrite here, and near to the dendrite, I have told you that there is an axon coming from here, just like here. So here we have seen that there is a dendrite here. This is the dendrite, and this is the axonite terminal from the previous neuron. So that is being shown here. And you can see that when this neuron is excitatory in nature, so that there is an action potential came here and neuron transmitters are released from here. And if the neuron transmitters are excitatory in nature, then essentially it will create an action potential inside the cell. So if this action potential is positive inside the cell membrane, naturally just above that, if you take that region, you can see that they are already at the resting membrane potential. But here it becomes a action potential. That means at a higher potential. So these two acts as a dipole, and there is an electric field originated between these two dipoles. Similarly, if it is an inhibitory postsynaptic potential, basically this action potential from the first neuron, when it reaches the action terminal, it generates neurotransmitters which are of inhibitory nature. So it creates a negative signal because the cell is now hyperpolarized. So it will become very negative, and compared to this negative thing, the upper region is positive because now this one is at minus 90 millivolt, whereas this hyperpolarized one is less than minus 90 millivolt. It can be minus 95, minus 100 like that, right? So that's why we put positive here and negative here. Again, there is a dipole with the polarization changed. The electric field is now in a different direction, right? So with this information, you can see that several of these neurons are kept stacked each other parallelly where the dendrites is upside down. Then you can see there are dipoles happening here, and these electric fields will get its effect in the scalp region. So these electric fields can get its effect in the scalp region by something like a vectorial sum, because there can be slightly difference depending on the spatial localization. So you have to basically add them all, and that will give me some signal, some effective electric field in the scalp, in which if you put an electrode, because of this electric field, this electrode will have different charge carriers passing through the electrode, which can be sensed as electrical signals in the brain. So this looks like a very random, noisy type of pattern that is coming from the brain, right? And from this EEG signal. So what is the forward model here? Forward model is nothing but all this mechanism with which these signals are being generated, like what we have already told. But of course, it is much involved, as I told you. It requires a lot of biochemical reactions, which has to be modeled. So all these things have been modeled properly. That acts as a forward model. So for a healthy patient, a healthy subject, it has got a nice structure, whatever. It's called the baseline or the basic structure. It's a very healthy for a healthy subject. But when there is a disease associated with the brain, essentially this forward model will get perturbed, so that some of these electrical signals will be distorted here. So now the question is, from these distortions in the measurement, which is my EEG, and along with the forward model. So what is the forward model? The doctors already know what should be the EEG of a healthy patient. That data is already there, right? So along with the forward model and the measurement, we will be able to diagnose the disease. And that diagnosis process is an inverse problem. That's how we have to do that. Now, again, you can see the Fourier transforms that we have already learned has got an immense potential here. So Fourier transform, we have seen that Fourier transforms can actually use this. So this is like a time domain signal. So y x-axis is time, y-axis is some voltage signal. So it's in the time domain. Now I have already told you, in order to extract certain features of the time domain signals, we can actually use Fourier transforms. So in fact, we can do a lot of filtering here to extract many features of this EEG signal with which we can actually analyze it. So Fourier transform is going to be a very important tool for EEG. Not only Fourier transform and different types of Fourier transform or different types of transforms like wavelet transforms and so many of them, or different variants of Fourier transform. Basically some sort of a transform to see it in a different way, in a different perspective, right? So these are the important tools. So this is how you have to see the signal processings are so important in bioimaging or bio measurements, that so many applications are there. Now, another thing that you want to tell about the engineering perspective of this problem is, so I have already told you that when you look at the sodium potassium channels, right? I have told that the sodium, there is an electrical potential associated with the sodium gate. So when the sodium ions move from a higher gradient outside of the cell to interior, essentially it involves the movement of the charged ions. So essentially it also causes a potential difference, and that potential difference is nothing but the Nernst potential, right? So individually, if you look at it, you can see the Nernst potential for sodium and the potassium, right? And then we see that in order to find the effective potential of the cell, that is the resting membrane potential, we need the Goldman equation, right? That we already see. I didn't do the derivations, but I have included those lectures in that slide. Now, if you want to model such a cell membrane to include this particular Nernst potential, we can consider a battery here, right? Equal with a voltage equivalent to the Nernst potential. So for the sodium gate, basically it is ENA, where ENA corresponds to the Nernst potential for the sodium. Similarly for EK for the potassium. Now, when the ions move across this terminal or across this gate, as I told you, there is a resistance offered by the channel depending on the molecular weight of the ion and the channel width. There is always a resistance. It's not a free pass, right? So there's a permeability, and from which you can calculate the resistance. So that resistance can go as a series to this particular Nernst potential. Now we can have several channels parallel. There can be a sodium channel, there can be potassium, there can be multiple sodium channels. So all these things can be parallelly arranged because all of them happen simultaneously, not one after the other. So if there are two gates and the sodium has to first pass through the one gate and then it has to pass through the second gate, then it will become series. But here the sodium can just pass simultaneously, potassium can pass, all are arranged in a parallel way. Simultaneously, the gates can be opened and closed. So this will become in a parallel configuration, right? And then what you have is a sodium potassium pump. So as I told you, the Nernst potential is basically a diffusion process. It will happen only from a higher concentration gradient to a lower concentration gradient, right? But sodium potassium pump is an active transport. It is always happen uphill. It can take things from a lower concentration to higher concentration, lower potential to higher potential, but you have to spend the energy. So the direction is different. So you can see that when the sodium channel is basically from outside to inside, sodium current as dictated here, we can see that the sodium current due to sodium potassium pump is basically from inside to outside. And so does the potassium case. So the polarity changes. You know why is it so, right? So this is how you can model it. And there is also can be a capacitance, which I have already told you. Because of the charge redistribution across the membrane, this membrane acts as a dielectric, and this can be a small capacitor. So there can be a capacitor across the membrane. So it comes in a parallel configuration. So with these electrical networks and this capacitance and resistance and potentials determined by the proper biochemical whatever values that we have to put, essentially this electrical circuit represents your tissue. It represents your cell membrane electrophysiology, right? It represents the activity, electrical activities being taken in the cell. There's a model for that. So if you know the Kirchhoff's voltage on law and the Kirchhoff's current law, essentially you can analyze this circuit and tell what are the outputs. How does it respond? Can you predict the action potential? how the action potential varies with respect to time. Everything can be done using circuit analysis, like what we do for engineering. And then we can say, we can interpret these results back into the cell signaling, right? So that's why it's a electrophysiology, right? So there are electrical concepts that can be applied here. So this is how, so these are the examples with which we can actually view this problem from the engineering perspective. So it is a really interesting subject. As an engineer, so, most of the things that you're going to learn is going to be quite handy in this bio instrumentation or electrophysiology or bioimaging, microscopy, right? There are many things that can be done in this biomedical engineering. So only thing is you have to you should basically do some courses in the department and then try to relating the things are important. If you learn things in a compartmental way, some courses, I mean, then that will not help. Then the idea is that you will do that course, you may do very well in the exam, and that's it. Nothing will come out of it. But if you just think beyond the courses, beyond your first year, second year, semesters, think beyond that, think beyond the courses, think beyond, even forget your department. What are you doing that you don't even worry about which department you are? If you can learn how to view this subject from that viewpoint, then you'll start enjoying things, you will start collaborating with many people, with many labs, and then you will see the how science works by looking at the things in a different way, and also relating many different disciplines from common perspective, right? So that's the idea. Anyway, so we are going to look at another concept, which is the ECG. So as I told you, there are other electrical signals that are coming from the body, which are ECG and also the heart sounds. So we will now look at what this action potential has to do with the ECG, and so for that matter, we should know what is basically this heart is, right? So we will first explain how does an heart works, and from that we will see how ECGs are being generated, or how to interpret the ECG, and what are the so we have already seen how to make an ECG in one of the videos, and what are these PQRST complex that we can actually now look at, try to relate it to the working principle of the heart, and how these things are related to action potential. So if you here, you can see that the heart has got two chambers, in fact 4 chambers, and it is divided into upper and lower. So the upper is called the atrium and lower is called the ventricle. Okay. Now in nutshell, what does our heart do is, heart has got to do 2 things. One, it has to pump the blood to whole body. Now what is the use of the blood? The blood carries the main use, what I would say is, it has to supply the nutrients and the oxygen to the cells. That's the main thing that it has to do, right? Why nutrients and oxygen? Because the nutrients and the oxygen will be taken inside the cell, which you already seen, and it will be converted into energy and stored in the ATP, right? So energy is what we require for all these things, and this energy is used by sodium and potassium channels for cell signaling. And this is exactly this energy is being used in neurons for cell signaling. And if there is no signaling, you are not a living organization, right? Living organ, right? So the senses are very important. One of them, not all. So the idea is that it's essentially the energy is spent in to sustain this life, to think, to see, whatever. I mean, if you don't think, what are you? Nothing, right? So energy has to be there energy has to be produced and stored, used and stored. It has to be used whenever required, or it has to be stored for future use. For that matter, you need oxygen and nutrients, and it has to be supplied to each and every cell. So there should be a circulation system. It is so, right? So there should be a circulation system, and that is being taken care by the blood. Now it has to be circulated. Why it has to be circulated? Because the things that are giving for storage of the energy can deplete. So when you spend the energy, that is gone. Now you have to produce more energy. If you want to produce more energy, you need more nutrients and oxygen. So that means there should be a supply depending on the demand, right? So every time, that's why there should be basically a circulation. So when it is depleted, it has to be again given the nutrients and the blood, oxygen. So essentially, the blood has to carry the oxygen and nutrients. The nutrients will be carried, taken from the gut, gastrointestinal tract, and then the oxygen will be taken from the lungs. That's how it works. Now, so the blood has to do two things. One, it has to, suppose the blood has got the nutrients and the oxygen, then this blood has to be pumped to the body. That's one thing. Second thing is the blood with oxygen and nutrients will be sent to the different cells. The cells take all the energy, nutrients and the oxygen, and it produces energy. And the rest of the blood, which is depleted with the nutrients and which is depleted with this oxygen, will be sent back, and that will be received by the heart, and it will be sent back to the lungs to get it oxygenated. Of course, it will be sent back to the guts as well for getting nutrients, right? So the heart has to pump the blood, oxygenated blood with the nutrients to the body. At the same time, it also has to receive the deoxygenated from the whole body and put it again back to the lungs so that it will get oxygenated. And that oxygenated blood will come back to the heart, which will be pumped to the body. So that is being taken here. But that these two processes are done by two parts of the heart, one by the left and other by the right. So you have to assume that somebody is looking at you from the screen, so that's how the right and the left are being notated here, right? So the left part, what it does is it receives the lungs oxygenated from blood from the lungs, and then it will pump it to the whole body. The right part of the heart, what does it do is it receives the deoxygenated or used blood from the body, and it will pump it to the lungs. So this particular route, it takes the blood to the lungs. It gets oxygenated, and it is coming back from the lungs to the right, to the left part of the heart, where it is pumped back to the body. So that's also so one part, the left part is called the body pump, and the right part is called the lung pump, if you want to say like that, right? This is essentially what it do. But then there are certain arrangements by which this is being done. Now, the idea is that there is a receiving unit and there is a pumping unit, right? In both the left and the right heart, there is a receiving unit and a pumping unit. So what does the receiving unit do? The receiving unit is basically on the left side is the left atrium, and on the right side is the right atrium. In the left side, the left atrium, the blood from the lungs is being received, and it's a chamber. Now, if you look at this particular heart, you can see this structure. You can see that it is called the muscles. Basically, these are all tissues, right? So it's a muscle called myocardium. Myo means muscle. Cardium means it's related to heart. So it's a thick cardiac muscle or myocardium, right? And this is a very thick muscle. Now, and it is divided into 4 chambers. Now you can see, if you see that this particular structure has got some AV1, SL1, SL2, and AV2. I have denoted here. These are called some valves. So these valves are being put in what is called a septum. Septum means you cut this heart along at the center. Suppose you cut it through the center. Then I will get a upper hemisphere and lower semi-hemisphere, right? You will get 2 hemispheres from the if you consider it as a sphere, you will get an upper hemisphere and lower hemisphere, and you cut it at the center. Of course, it is not a sphere or anything. I'm just telling you to explain it to you. Now, the center part is basically called the septum, which separates the upper from the lower. And center part is basically has got a thin membrane on which these valves are being fixed. AV1, SL1, SL2, AV2. Right? So there is a thin membrane over which this upper hemisphere and lower hemisphere are fixed to form the sphere, which forms the heart, right? And it is further divided into 4 chambers, right? Now, what happens is when the blood flows to the left atrium, essentially the blood will get accumulated in this left atrium, so that when the pressure increases, this AV2 valve opens and the blood will go to the left ventricle. So all these valves, these are called valves. These are not a opening. It's a valve. Valve means it will open only in one direction. Okay. So when the pressure in the left atrium increases, all the blood will come to the left ventricle because of the pressure, right? So it will start filling because this valve will open from atrium to ventricle. That is the direction of this valve, not from the other way around. Suppose the left ventricle pressure increases, this valve will shut close. It will not open to the atrium. It will just keep closed. But if the pressure in the atrium increases, this will get open to the ventricle. So the blood can come from atrium to ventricle. So the idea is that the blood from the ventricle will not go to the atrium, left atrium, but blood from left atrium can come to the left ventricle. And similarly for all other valves. So I have shown the directions in which the valves are there. So in that direction only the flow can happen. If the blood try to come down, say for example in the SL2, if the blood try to come from top to bottom, SL2 will close and it will not allow it. That's the idea of valves. So you can see that in the left atrium, the blood will, due to pressure, the blood can come to the left ventricle. And in the left ventricle, it will pushes this blood so that SL2 will open and it will go through what is called a big vessel called aorta. And this aorta will branches out to arteries, which I have already shown you in one of the slides. And the arteries will branches out to arterioles, and the arterioles will branches out to capillaries. And from capillaries, they are in the vicinity of the cell from which they can take the oxygen and the nutrients. That's how it works. Similarly, the blood from the upper part of the body comes from what is called superior vena cava. Lower part of the body comes from the inferior vena cava, and both will merge to and reaches the right atrium. And again in the right atrium, when the blood pressure increases, because all the blood are coming here, at some point the pressure increases so that AV1 will will open, so that all the blood will start filling the right ventricle. And then what right ventricle do? It will push it. So it will push this blood so that SL1 will open, and all this blood will goes to the lungs. So pulmonary means it has something to do with lungs. So this is pulmonary artery, means this blood is being taken from the heart through this pulmonary artery to the lungs. And in the lungs, it will get oxygenated. Now, once it is oxygenated from the lungs, there is another way which is called pulmonary vein, through which the blood from oxygenated blood will fill to the left atrium. That's how the circulation continues. So the aorta will take all the things to arteries, from arteries to arterioles, arterioles to capillaries. In the vicinity, you have venules. Then there are veins, from the veins through inferior and superior vena cava, and then it reaches the atrium, from atrium to ventricle, and ventricle to pulmonary artery to the lungs, and from the lungs to pulmonary vein, and from the pulmonary vein to left atrium, and so on. So this keep on continuing, right? So this is how it works. Now we can see that the muscles in the left ventricle is little bit thick, whereas the muscles in all other parts are very thin. So the idea is that this left ventricle is the body pump, I had told you. So it has to pump this blood to the whole body. And you know, heart is just on the left side, roughly off-center to the left side of your things, very small one compared to the body. And this body has to get the blood with some pressure. So you can imagine how pressure you want. So this guy has got a very thick muscle so that it can actually push it. So pushing is basically nothing but like a balloon. So you assume a balloon filled with water and you want to basically push it out. So what we do, we will press with both of the hands from all directions. We will press the balloon so that the water can just gush out, right? So that is exactly what is being done for in all the cases. Essentially, these muscles contract and this muscle will push from all the sides so that the volume decreases but pressure increases, right? Because when the volume decreases, pressure increases here, and that pushes the blood to the aorta, right? In fact, it is not just pushing from all the sides. It also give a twist. So in heart, you can see the videos. Basically, it is not just compressing all the muscles to reduce the volume, but it's also there is also a twisting action to give extra push of the blood from the left ventricle to the aorta. Similarly, the right ventricle also do the same thing, but it doesn't have to do as vigor as the left ventricle do, because it has to just push the blood from the ventricle to the lungs. That's all, right? So that's where things are being evolved, so that ventricle has got more thick muscle and the other has got thinner ones, right? So this is how it works, and the pressure, the left ventricle will push it As I told you, the left ventricle will push the blood through the aorta, which will give me a very high blood pressure. And that part is called the systole. So in blood pressure measurements, there are two things. One is called systole and diastole. So when it pushes, when left ventricle pushes the blood, that part is called the systole of high blood pressure. And when it relaxes, there is the lower blood pressure called the diastole. So these are the two things that we measure when you use a sphygmomanometer to basically measure the blood flow, right? So yeah, so this is essentially the working of this heart, right? So this is how it works. These are the things that I have already told you. You can read it by yourself. These are again, these things I already explained. Yeah. Yeah, these things also I have explained. Yeah. So this is something from this arrangement we can learn what are called the heart sound. So as I told you, okay, so there is one more catch here that we have to think. So as I told you, this atrium, the left atrium and the right atrium are receiving the blood. So the blood will come from lungs to the left atrium, and the blood will come from the body to the right atrium, right? And now this atrium, when its pressure increases, it will move to the right ventricle. Similarly, blood from the left atrium move to the left ventricle, and it is pushed either to the lungs and to the whole body. That's how it works. But then there is a catch here. Suppose the left atrium, blood is filled in the left atrium, but at the same time, the left ventricle tried to push it. That's not a good idea, right? Because left atrium is filled with the blood and the pressure is started increasing. The blood is just started coming to the left ventricle. It has not filled the left ventricle. It is just coming because left atrium is doing the job of increasing the blood, receiving the blood, and its muscles are trying to push it, and slight push of muscles, and the rest is because of the gravity and the pressure. The blood from the left atrium is started coming to the ventricle, but the ventricle is not waiting enough. It started just pushing it. Is it a good system? No, because you have to basically, if I will design a heart, I would design it in such a way that basically you have to wait till the left ventricle is filled with the blood, and then only I will try to push it. So the working, there should be a time coordination between the working of the upper heart and the lower part. So we have to wait till the left atrium do its job, so that all the blood from the left atrium fills the left ventricle, and then only left ventricle has to push it. Similarly, the right ventricle has to wait till the blood from the right atrium fills the right ventricle, and then it has to push it. So the pushing action or the pumping action of the ventricles should be delayed compared to the pumping or the pushing action of the atrium. You have to wait a little bit, right? So that's a very important parameter that we have to look at. So you have to little bit wait till this is pushed up, this is pushed. So first that right atrium will push the blood to right ventricle. That time right ventricle will wait, it will not pump. Similarly, the left ventricle, and once it is filled, it will push it. So there's a coordinated action that is happening that you have to keep in mind. We'll come back to this point later. So with this one, we'll see certain heart sounds. So I told you we are looking at certain signals coming from the body for diagnosis purpose. And one such sound is a heart sound. So heart sound is basically coming from these valves. So when you open a door, you will never hear a sound. But when you close it, you will hear the sound, right? So similarly, here the heart sounds are mostly created by when the valves are shut down or closed, not while opening. So here you can see that when the ventricle, so here let me use the same figure so it will be easy. So if you look at this ventricle, so when the ventricle pushes with high pressure, this SL1 valve will open. As I told you, it is designed to open to the aorta or to the pulmonary artery. So when right ventricle and left ventricle contracts or it compresses, it creates high pressure, and both these valves will be open so that the blood can be pumped to the body or to the circulation or to the lungs. But at the same time, when the pressure increases in the ventricles, AV1 valve and AV2 valve will be closed, Because these are valves, it cannot open to the atrium so that it will shut close. So this closing of AV1 and AV2 creates a sound because it is shutting down as it's closing. So that creates a sound, and that sound happens when there is a systole or there is a high pressure that is happening. So when there is a high blood pressure, because of the ventricle pushing the blood to lungs or the body, that time AV1 and AV2 will close, and that is called the first heart sound. That is called lub. So this lub dub that we see with this stethoscope, essentially that lub is because of the AV1 and AV2. Right now, what happens is after pumping this, the blood, now the atrium has put all the blood to the ventricle has pushed it to the body or the lungs. Now atrium doesn't have any blood, so that the blood from the circulation is now started filling this one, right? So now ventricle has to wait. So ventricle will expand so that the pressure here increases. When the pressure here decreases, when the pressure here decreases, the aorta and the pulmonary artery still has the blood. So that pressure pushes this SL1 and SL2 back, but it cannot come to the right ventricle. So it has to shut down both of these. So when the SL1 and SL2 closes, because of the back pressure from the aorta and the pulmonary artery, where the ventricle is now in the low pressure, essentially this is the period of diastole. And that time, because of the shutting down of SL1 and SL2, you will see another sound, which is called the second heart sound, which is called the dub. So lub dub is basically because of the heart valves closing. One is this AV valve which closes, and the another is a SL valve which close. So there are names for this AV and SL. I really don't care about the names. Just there's a atrioventricular valve and also so many terminologies are said, so don't worry about it. It's basically the working that matters for us. So basically, it's like a mechanical pump with a lot of valves, and in fact, we can make an artificial heart to mimic this function, right? So this is something that you have to keep in mind, and this lub dub can be basically measured with a stethoscope, and from this from the frequency of this lub dub, basically you can infer the status of the human heart. Right. So again, there are forward model, how this sound is being generated. You know, what should be the heart sound pattern for a healthy patient. You know, that is a part of the forward model. The measurement is taken by a stethoscope, and then it is fed to, it is heard by a doctor. Doctor knows what is a forward model. He's getting the measurement. And from this, he can see whether the measurement is deviated from the actual one. That means from the actual forward model or the actual healthy subject's value. If there is a change, he can infer, okay, see, look, there is a change. There is something not good, right? So there is an inverse problem happening in the doctor. He's taking the diagnosis, and that's how it works. Now now up to this point, we just said the heart is pumping, but we never said how actually these muscles contract. So for the balloon case, I am pressing it with my hands, but who is pressing the heart, right? We don't have to press it always, right? It's doing its job. So how does it do? So essentially, here comes the electrophysiology. Essentially, the electric signals, the action potentials that we have learned is essentially what actually causes these muscles to contract or expand. So you actually, there is one when you fed certain muscles, motor neurons, basically with some certain type of electrical signals, essentially it can cause, in fact, not motor neurons, sorry, it's basically the muscles. So when it is fed with some electrical signals, essentially these electrical signals can cause the muscle to basically contract or expand. So there is a mechanism for that. It's basically physical movement. Physical motions can be triggered. Mechanical movements can be triggered by these action potentials in certain type of muscles. And that is being dealt in one of the chapters in Guyton. You can have a look at it, but I'm not going to take those parts. But assume that these electrical signals can actually make these heart muscles contract or expand. So compress or expand, both can happen by these signals. That's exactly what we are going to, we assume that. And now we are interested only in what is the nature of these signals and how it is being created. So if you look at this particular figure, you can see that in the first figure also I have put it. So there is a small sort of a plug here, which I call it SA node or sinoatrial node. So this is a region very near to the superior vena cava inside the heart muscles. It's a special region in the heart muscle. So the sinoatrial node has got an ability to generate certain electrical signals by its own. It's an inherent signal, so sort of a signal generator or an oscillator in the engineering viewpoint. So this creates a series of pulses, and these pulses is passed to the muscles, this myocardium, these outer muscles, and these muscles will contract or expand or compress, get compressed based on these electrical signals. When the electrical signal comes, it expands. If it is absent, it goes back to the previous position. Right. So that's how this. So when we compress the balloon with the hands, that action is being taken care by these electrical signals with a coordinated effort with the muscles. Now the question is, these signals are being connected as taken by some fibers which are attached to this myocardium or the cardiac muscles, and some of them, so it has to be branched to whole thing. So it has to be connected to atrium as well as ventricle, right, that we are seeing. But there is a catch again here. I have already told you that the ventricle has to do the pumping action only after a time after this atria pumps, right? There should be a time delay between the atria and the ventricle contraction. When atria is contracting or when atria is pumping, ventricle should not pump. It has to wait till the blood reaches the ventricle, and then ventricle has to pump. That time atria has to remain idle, right? So there's a time delay between these two. So naturally, the electrical signals which are causing this contraction of these muscles, these electrical signals should also be delayed. So the signals that are fed to atria should be a little bit ahead compared to the signals being fed to ventricle, or there is a delay for the signals in the ventricle. This has to be ensured. And there comes two concepts. One is there is a relay mechanism for these signals here. So the signals that are given to the lower part, that means the ventricle, is actually being relayed through what is called an AV node, atrioventricular node. So this causes these electrical signals to have a small delay. So if this particular SA node creates a signal at say 0th time, that means at the origin, this guy will pass it after a delay. Say for a few microseconds, it will be delayed and then it will come to the lower part of the ventricle. So this will ensure that when a signal is generated in the SA node, the atria will first contract, and then it will fill all the blood to the ventricle. By that time, this signal has not reached the ventricle, so it will not contract. So it will wait. When the signal reach, it will pump. So by that time, the atria has done its job, right? So that's how we have to view that. So this is one important thing. But then another thing is that although there are branches that are going to this myocardium, you may think that these signals can actually pass through the myocardium and reaches out through outside, right? Through the surface, through the surface, it can reaches the ventricle so that it can cause contraction. But that will not happen because of the septum. So as I told you, you consider this as two hemispheres. Between these two hemispheres, there is a septum, and in that septum, that means septum means there is basically something we separate. That membrane, in that membrane only these valves are sitting. That membrane is not an electrical conductor, and that membrane actually divides these two hemispheres physically. So any signals that being transmitted in the upper atrium is not being passed to the lower ventricles. So that these signals will reach the ventricles only through AV node, and AV node always create a delay, so that these coordinated effects will work. So this is something which you have to keep in mind. Now what? So these are the things that I already told you. So now we have to look at where are these signals coming from and what are they do. So first, let's look at the action potentials being generated in the ventricular muscles. So if you look at these ventricular muscles, which causes the pumping action, You can see the action potential is basically it's around 90 millivolt resting potential, like what you have already seen. So these cells, these muscles also has got the cells which has got the sodium, potassium gates, everything arranged there, like what you have already seen in the last lecture. And then there are here, it is not just the sodium gates that works, a calcium gates also works. So the calcium is also abundant outside. So when some due to some action potentials, basically when the sodium and the potassium, when the cell potential increases, interior increases, both the sodium and the calcium channels will get open. So both the sodium and calcium channels is taking part in the depolarization process, so that it will go from -90 to +20 millivolt. And it's actually, there is a plateau here. If you remember that our earlier action potentials were actually coming down, so the repolarization happens very fast. But here there is a plateau. The plateau is because of the calcium channels are a little bit slow. So even if the sodium channels will stop because of inactivation gate, calcium will start flowing from outside to inside still, so that the depolarization is kept little bit over longer time. That means it is almost in the positive for longer time. And this is very much important for heart muscles, because for heart muscles, the depolarization is the pumping that is required for us, because you should push the blood forcefully as you can, so that the blood reaches with enough pressure to the whole body. So basically you have to uh, have this depolarization for a longer time, so that we can have this pump efficiently. So calcium is basically helping for that matter. So it will have the plateau here because of the calcium channels, and then the repolarization happens because of the potassium, and it comes down. So this is how it works. So sodium and calcium is acting. Calcium is helping to retain the polarization for depolarization for longer time, which is required for the heart muscle. Now the question is, this is fine. This is actually the action potential of the muscles. But who is initiating this? This is actually a signal coming from the SA node through the AV bundle. Through the atrioventricular node to the ventricle. But then where is the signal coming from the SA node? Because this heart is always pumping. It doesn't. No, we don't have to initiate it, right? So there is some signal generation that is happening with respect to time, without stopping, like an oscillator. Oscillator is something which always generates a signal, right? You switch it on, it will keep generating the signals, right? So where is that coming from? So that is happening in the SA node. So in the SA node, the idea about SA node is that this SA node also has got sodium channels. It has got calcium channels. It has got channels which can pass both the calcium and the sodium, and it has got potassium channels. Now, if you look at here, you can see that the sodium channels in the SA node muscles, that means atrial node muscles, it has got a special property. They are leaky. They are leaky in the sense that the channel is slightly open, so that the sodium can actually leak from outside to inside. As I told you, sodium is abundant outside. So there is a natural diffusion. If the channel is leaky, it can keep on coming in a slow quantity to inside. So when the sodium keep on coming in a slow quantity to inside, the cell potential will keep increasing. See, if this is the resting membrane potential, roughly around 50, it will keep slowly increasing. Why? Because the sodium channels are leaky. So the cell interior potential is keep increasing like a slow slope. And when it reaches -40, then this is a threshold where the calcium sodium channels can open. But as I have already indicated you in the previous lecture, that when the sodium calcium, so sodium channels will get reactivated only when the resting membrane potential reaches -90 millivolt. But here, since the sodium channels are leaky, it can never get activated because it is not at the resting membrane potential. The inactivation gate is always closed. So sodium channels do not have a role here. But because of leaky sodium channels, when it reaches -40, an action potential can be triggered using the calcium sodium channel. So a calcium sodium channel is a channel where calcium and sodium can go inside. So when the membrane potential reaches -40, the calcium sodium channel opens and a lot of calcium and sodium start coming inside. That will increase the voltage to a +20. But then at some point, the calcium sodium channel will stop. But then that is a slow process. That's why there is a plateau here. So the depolarization is slightly higher, and then the potassium start coming inside. And when the potassium starts coming inside, calcium and sodium channel will get deactivated. So potassium keeps going out. So repolarization happens and the potential start coming down. And at some point when it crosses -40, the potassium channel stops. Because the cell potential has gone below -40, so it has to stop. But then it cannot go -90 because of leaky channel. So again, the sodium leaky channel will start pushing it from this point again to -40. So the leaky channels will never allow the sinoatrial node to rust. It will never be at resting potential. It will keep pushing it to -40. At -40, an action potential comes, and then repolarization push it down to some value. But again, the leaky channel will again push it back to -40. So it will never allow the SA node to rust. It keeps creating disturbance, which forces the SA node to create action potentials. And these action potentials are being transferred to the atrial muscles and the ventricular muscles, which will respond to this using ventricular muscular, this action potential that we have seen here, right? So this SA node acts as an oscillator. So you can see that. So it acts like an oscillator, with where we can see because of this leaky channel, it keep producing these signals, right? And this causes the heart to. So when it stops, you are dead, right? So this is essentially how the heart signals are being generated in the SA node, and this is being passed to atria and ventricle, and it causes the muscle to contract, and that is the pumping action. So that the blood, when it contracts, the pressure increases, volume decreases, the blood will start flowing to the whole body, right? And these signals, this ventricular action potential signals from different parts of the heart muscles will propagate through the body, and it is available to be measured at the chest. And that is exactly what we measure as electrocardiograph. So if you look at the electrocardiograph, we have got three things: one, P, and then QRS complex, and then the T wave, right? So what is P? So P is a wave when the atria depolarize. That means, as I told you, the atrium has to basically contract to pump the blood which is received by them to the ventricle. So that contraction, that signals will be coming from the SA node to the atria, and it will contract. So that particular signal P corresponds to that depolarization or the contraction of the atria, and that is this P. Now, once the atria contracts, the ventricle will wait for some time because the signal has to reach by a delay by the AV node to the ventricle, and then the ventricle will contract so that the pressure increases and we get the systole, or the blood is pumped. That time you will get another signal which is called the QRS. So this corresponds to the depolarization of the ventricle. Now, after some time, what happens is the repolarization of the ventricle happens. The repolarization of the ventricle means the ventricle contraction stops, right? So that time there is a repolarization happens. There is a signal coming corresponding to that repolarization, that is called the T wave. The repolarization due to atria is also there, but that is actually more or less coinciding with the QRS complex, but of small magnitude, so we won't see it. Right, so this is how the PQRST has to be interpreted, and this signal is what we have actually measured using the experiment, right? So those details are you can find from that particular video, or if you are interested, you can come to the lab. We can arrange some experiments at some point. Yeah, so these are the things, and this is something called cardiac cycle. So all these things are being explained here. So for example, if you look at the systole, so systole is the point where the high blood pressure comes. So that time the atrioventricular valve shut down. So that causes the first heart sound that is shown here. But at the same time, systole happens at the beginning of the depolarization of the ventricle. And that is exactly where the QRS complex comes. See? And when the QRS complex comes, what happens to the ventricle? The ventricle has to contract. So its volume should decrease. That is shown here. So when this systole happens, the volume is decreasing. See? And when the volume is decreasing, the ventricle will reduce the volume so that the pressure increases and pushes a lot of blood with high blood pressure to the aorta, and that's why the aorta pressure is increasing. You can see here. So this is how you have to read it. So if you can read everything, how these things are happening, essentially it means that you understood how this electrical activation of the heart muscles is coordinated with this pumping action, right? So this can be read. The whole thing that we are already told is what do you call it? It's basically put it into this particular form, like a cardiac cycle where you can learn it. So this is essentially what I have already told you. All these informations are here. You can learn by yourself. So that's all. So if you have any questions, you can ask. Otherwise, we can stop or you can leave.
So,
Hello. Any questions?
so
Yeah.
can you please explain the cardiac cycle on the previous slide? Like, what part you said?
Which one? Last one.
Yes, this one.
Yeah. So here, if you see that PQRS complex, so first what happens when the atria? So you don't have to. You can convince yourself. I'll just give a hint what is going on. So when atria contracts, basically that is the point where the P wave comes. So this P wave comes when the atria contracts, right? So that time the atrial pressure will start increasing. So you can see the atrial pressure increases because depolarization causes the atria to contract so that it will push the blood from the atria to the ventricle. So this pressure increases just after the P wave. Then what happens? The ventricle will not work that time because the ventricle will get the signal after a delay. So after some time, the ventricle will get the depolarization signal at Q. So the moment Q comes, basically you can see that the depolarization is going to happen for the ventricle. That's why the QRS complex comes. That time the ventricle has to contract. So when ventricle contracts, essentially its volume should decrease. But at the same time, this blood is now pushed to the aorta so that aortic pressure should increase. So this is called systole because systole is the point where the blood pressure is increasing, right? And then this process continues. Then depolarization happens like that. And the first heart sound comes when the ventricle is being contracting, and second heart sound comes when the semilunar valve, this SL1 and SL2 valve, which connects the ventricle to the aorta or the pulmonary artery, that shut down, right? That will happen at the end of the high blood pressure. So that will happen here, second one. This third heart sound is basically the murmuring sound, basically from the turbulence of the blood inside the thing. So it's very difficult to get as well in a usual stethoscope. So these are the two things that we measure, right? So yeah.
The increase in the electrocardiogram after S, in that case, at the aortic pressure and atrial pressure, why is it still decreasing?
Sorry, I'm not. Your voice is breaking. Can you slowly tell me once again what is that?
So in the bump in the electrocardiogram after S, the increase. So in the corresponding position of aortic pressure and atrial pressure, why isn't that also increasing suddenly?
You mean in the ECG, right? You are talking about T wave, I guess.
Yes, sir.
Yeah, T wave is basically the signal corresponding to repolarization of the ventricle. So when the ventricle repolarizes, right? So that corresponding to that part, there will be a wave coming from the body. So T corresponds to the ventricular repolarization. So when it comes, it means that the ventricle is going to have this low volume. So the ventricular volume is coming low, so that aortic pressure will comes down. So it is the starting of the diastole. So that's the idea. And at the same time, there can be a repolarization from the atria because we are considering only depolarization of the atria. So repolarization of the atria is also there, but unfortunately that is being embedded inside this PQRS. It's basically coinciding with the QRS time. So you need to have a higher resolution ECG with which we may be able to differentiate it. That has to be extracted from the QRS complex. So it's not very easy to extract it from here. So we look only for P, QRS, and T. And look, this shape is also a very confusing stuff. So you may think why it is going down and then it's up, and why it's going down and there is a peak. So all these things can actually change when you change the detector position. So there is a hell lot of theory on how to put the placements of the detectors in the chest. So you can see if you look at the guide, and there is a chapter on vectorial sum of this EEG. Wherever you put it, you can actually do some vector sum to get how the signals look like. Right, these are potential differences between 2 points. So the leg is usually in one configuration, the leg is usually taken as the ground, and you have got chest leads in somewhere in the chest, and then you have to do a vector sum to get all these things. So all those details I have skipped here. This is just a very quick introduction how these signals are being originated at the cellular level, and what is its role in getting physiological signal at the body. But why this shape is of this form, whether this shape will change with respect to detector position in the chest, how do you interpret it? It's a completely different story. So it has to be analyzed in a different way. Some of the ideas are given in the guidance, and there are many other textbooks which will tell you how it has to be done in a rigorous way.
Yes.
Yeah. So anything else? So the structure of the exam, I will inform you. Mostly, I will try to minimize the questions from the last few lectures or the second tutorial. I'll try to minimize it, and I can also give some practice questions. I will try to post this today itself, or maybe tomorrow early morning. I will do it today itself. That's better. The video of this lecture will be posted within next 1 hour. Questions will be objective type. I don't know the exact marking how I am going to do that. But you will have enough time to handle it. It's multiple questions, 4 choices or 3 choices, only one correct answer. There won't be any negative marking. There won't be any quantitative type questions. You don't need to even do any numericals. It's mostly logical or qualitative type. It's just look at your understanding. So if you can watch all the videos, that is enough. Nothing else is required.
So what is the duration of the test?
Duration, today you might have received the mail, right, from Ambarish. I think he has sent it, I guess.
There's an
So
error
it's
that,
3 hours. Huh?
Sir, I think there's an error in that the time duration says 232.
Your voice is breaking again. Can you repeat? Let me see what is the error. You said some error in that one, right? That's what you said.
Yes, sir. The time duration written in the mail states 2 hours 15 minutes. But the exam duration states 2:30 to 4:50. So that's 1 hour 45 minutes.
Okay, that I don't know. 2 hours 15 minutes. 2:33 hours for PWD. 2 hours 15 minutes for all students. So it says that 2 hours 15 minutes for all students and 3 hours for PWD category students. That's what it says. So if you count it like that, even then it's wrong.
Yes, sir. It states the time duration states 2:30 to 4:50.
To 3:30.
2:30. 2:30.
2 to 3:30. Yeah, that is true, because now it is only 1 hour, right?
1 hour 45.
1 hour 45 minutes, not 2 hours or 15 minutes.
Yes, sir. So which one is correct?
I will get back to you. I will talk to him, and I will, if there is some issue, I will tell him to correct.
Okay, sir. Yes.
Yeah, I will do that. Yeah. Yeah. So if you have any doubts, you please post it in a Google, some forms that I have circulated in the Moodle. I think I have posted it in one of the announcements. If you have any doubts, just type it. I will try to answer it, whatever. But please don't put it to Sunday night or something. I mean, if you have any doubts, please put it as early as you can.
Sir?
Yeah.
But like mostly it will be like 25 marks or 25 questions only, right? Or like maybe more than 1 mark.
Voice is breaking again. I'm not able to hear you. Can you?
Can you hear me now?
Yes, please.
Yeah, I was asking that the number of questions would be 25 only, like 1 mark each, or can it be more?
No, I don't think so. That will be too much for me. I mean, it will be something like 2 marks, 3 marks, or 5, depending on the question.
So like the total marks will be 25 only, or will it be scaled later on from?
No, it will be 25. That's easy. I didn't think about it. That's but I will give enough questions from couple for first 2-3 for first four lectures or first 5 lectures, or maybe 4 lectures and 1 tutorial, and couple of one or two questions from last 2. Or maybe I will try to avoid it, depending on I have to see that. But I cannot say I avoid it, but I will try to decrease the number of questions from the last lectures. That I can guarantee.
Oh, okay.
Yeah.
Thanks.
Yeah. Okay. So any other questions? Any queries regarding the exam or anything? If you want, or you just send me a mail, I will respond to it. So the last, okay. So for the last batch, I have already uploaded the video. This one I will upload it soon. Okay, so I think there are not much questions. So let me so just post the questions in the Google form, and that's it. So don't worry too much about the exam for my module. It will be quite easy. Only thing is you spend some time looking at the, I mean, watch the video. That's enough. Nothing else is required. Just watch the video. You'll be able to answer, right? Okay, so then we can leave. Yeah, thanks.
Thank you, sir.
Thank you, sir.